Good morning, good afternoon, good evening, and thank you for joining us in this fifth seminar for the ninth series of International Plant Breeding Seminars, hosted here from North Carolina State University. Today we have the honor of having one of our faculty presenting the seminar, Dr. Dilip Panthee. He's a professor here in horticultural science, and he actually works in the western side of our state at Mills River Station. And his research focuses on developing tomato lines and varieties that are adaptable and disease resistant and high yielding for the fresh market. And also he studied the inheritance of several relevant traits for the breeding of tomatoes. He before joining NC State, he got his PhD and did a postdoctoral time at the University of Tennessee in Knoxville. And before that, he got a Master of Philosophy from the University of Birmingham and Reading. And before that, he was in Nepal at the University of Tribhuvan, where he got his bachelor and also worked and taught there. Okay, Dilip, it is a pleasure to have you with us. Let me stop sharing here and give you the floor. Go ahead. Stop sharing.
Right,
You are set. You're set.
Thank you, Carlos, for nice introduction. Can you see my PowerPoint?
Yes, you're ready. You're set.
Okay, very good. Good morning, good afternoon, and good evening, wherever you are. I am so honored to present my research this morning. I'm going to talk about breeding tomatoes for foliar disease resistance. It does not move. That's interesting.
Click the screen and then try move.
Yeah, okay. All right. Yeah, good. Thank you. Yeah. Okay, as you know, economic importance of tomato. Many of you already know that tomato is very important vegetable crop, second to potato. If you look at the global production, China is the #1 producer, followed by India and USA. On the right-hand side, as you can see, this is the top 10 producer at a global level. And if you look at the total area under production, there is about 4.6 million hectare in the world, producing about 130 million metric ton. That shows the overall economic significance of the crop at global level. In North Carolina, though, it is produced only in about 175,000 hectares, and it is about $1.4 billion industry. Majority of it is produced only in 2 states, California and Florida. North Carolina, where I am located, contributes only about 3% of total production at national level, which is about $32 million industry. North Carolina, on left-hand side, as you can see here, as you can see on left-hand side is the map of North Carolina. Tomato production is basically located on western part of the state. These are the counties on right-hand side, which are magnified. These are the counties where the tomato is produced to a large extent. Although it is produced in Piedmont, which are differently colored in eastern side and in Piedmont. although it is produced to some extent in all regions, but it is basically produced in western side. That is why I am located here in the west. Overall, breeding objectives of the program are basically categorized into two. One, developing varieties, as you can see in all breeding programs. In developing varieties, my program is also focused in addressing or developing varieties related to disease resistance, food quality, and heat stress tolerance. As a public breeder, we are trying to develop varieties for bacterial, virus, and also fungal disease resistance. And for food quality, like food lycopene content, food flavor, and other related food quality traits, and heat stress tolerance in abiotic stress tolerance, which is the priority. Okay? And also, there is a main emphasis in molecular breeding, particularly developing molecular marker, and use those molecular marker in marker-assisted selection. But in the interest of time today, I will be mainly focusing in foliar diseases, particularly in fungal disease and bacterial spot and early blight, late blight, and septoria leaf spot. As shown here in this slide, late blight, early blight, and septoria leaf spot and bacterial spot, if we do not tell general people, these diseases are kind of confusing. Late blight is very obvious, as you can see, disease plant and resistant plant, or susceptible plant and resistant plant. But rest of the diseases are sometimes very confusing. In fact, that is the case when we go to the field, and particularly it becomes confusing when we are scoring and so on and so forth. But overall message of this slide is that it is not only confusing, but these diseases cause a significant crop loss in the program. First, bacterial spot. Bacterial spot is a major disease in North Carolina, and I believe that this is also a problem in tomato producing area around the world. This is caused by a Xanthomonas species, Xanthomonas complex, I would say, because there are multiple species that they cause this problem. As you can see here in this one, there are 4 different species. Among those species, there are races like T1, T2, T3, and T4. And if there is conducive condition, particularly hot and humid condition, this disease can cause up to 66% loss. Range-wise, there is about 17 to 66% yield loss. It can affect all parts of plants, including petiole, as you can see here in this slide, all the way from flower, fruits, stem, and foliage, of course. Considering the effect on all parts, means including fruits, it affects not only the yield loss, but also the yield quality. It is not only quantitative loss, but also the qualitative loss in fruits. Considering this situation, we developed population and started mapping to investigate the genetic analysis, map the QTL, so that we can develop the molecular marker, and also at the same time we can develop the lines, resistant line for that. and graduate students were involved in this project, and we evaluated in multiple locations, as you can see here. And, this slide shows the distribution of the disease in multiple locations, as you can see here. This is it's on left-hand side, as you can see here, it shows the simply correlation from multiple environments. Very simply, larger the size of the size of this one, greater the magnitude of the correlation, as you can see here, and also the star shows the significance. On right-hand side, it shows the principal component analysis based on the angle of any two location or any two variables. We can look at the principal component, level of principal component of any trade, or in this case, we were trying to look at the location, how much variability was there. This slide shows the heat map heat map of the in different chromosome, and also number of molecular marker in different chromosome. Say for example, on x-axis it shows the number of markers, and also on y-axis here it shows the number of molecular markers, whereas on this side it shows the chromosome number, and on top it shows the chromosome number. Based on that, which chromosome number has how many markers, you can look at that. Say, for example, this one has like 400, 200, this one has like 600, and this one has 800, so on and so forth. This slide shows the distribution of molecular markers, and its a length, chromosome length, and average molecular marker interval, and the length, again, total length in linkage analysis. We performed the QTL analysis associated with bacterial spot resistance. The first column shows the environment where the population was evaluated. Second one shows the chromosome. Third one shows the molecular marker. In each, you can see these two molecular marker. These are flanking molecular marker associated with the QTL. And the third one shows the map position. And next one shows the physical position, load score, and additive effect and phenotypic variance explained by that particular QTL. Obviously, there were multiple QTL detected. This slide shows the QTL detected from chromosome number 2, 6, 7, 11, and 12, as you can see the position on various chromosome number. On right-hand side, it shows the contribution from homozygous, let us say, homozygous dominant, homozygous recessive, and heterozygous contribution of those alleles towards resistance. Okay, from different when they were evaluated in different locations. This information has already been published in this publication, as you can see on the bottom there. And the same study was also verified in an independent population, and again the QTL were detected from chromosome number on chromosome number 2, 6, 7, 11, and 12. And this is also published in the same publication. It was same publication as before. Similar to that one, we as a follow-up study, we performed the RNA-seq analysis. For that, we evaluated multiple lines, and to see the level of bacterial spot resistance. As you can see in this graph, on right hand side, there were one was it shows the AUD DPC value evaluated over days after planting. Some of the lines were resistant, as you can see here, like PI 274.3, and one was kind of intermediate, and whereas the other one was susceptible. And using these three lines, although there were some other lines also, we decided to choose those three. And those three lines, when we perform the cluster analysis, those three lines cluster very well. As you can see, PI 270, 443, all lines, collected at different time points, they clustered together here, whereas NC 714, which was susceptible, it clustered together here, whereas NC 1 CLBR clustered together again here. This slide shows the number of genes differentially expressed here. On left-hand side, blue bar shows the upregulated number of upregulated genes, whereas red bar shows the number of downregulated genes. The first bar is PI 274.43. Second bar shows the NC1 CELBR, which is intermediate, and the third one is the NC 714. Again, overall, number of downregulated genes on right-hand side, the circle shows the number of downregulated genes, which is like 362, and upper circle shows the number of upregulated genes. Without going in detail, all that information is published here in this publication. And using that information, we also mapped. What is interesting is, the same information was also mapped on the same chromosomes that we reported before. That is chromosome number 2, 6, 7, 11, and 12. Although not exactly, but some of the information was common. In the same study, we also performed the pathway analysis using these upregulated genes and downregulated genes. Obviously, these genes are involved in various pathways, like they are defense related, as we know. These genes are involved in different, whether like EMAPK or transcription transcription factor, so on and so forth. All those bunch of genes, which we were interested in those pathway also, so that we can identify these specific genes. Next, early blight. We already know that early blight is the major devastating disease, fungal disease, and it is there are multiple nomenclature wise, there are multiple species like Solani or alternata, so on and so forth. Anyway, mainly symptom wise, it has light black spot when we go to the field, but it has like concentric ring when we go to the field, as you can see here on right hand side, concentric ring. And the size wise, it has slightly bigger size compared to septoria leaf spot, because sometimes it is confusing between septoria leaf spot and early blight. This concentric ring is very typical of early blight when we go to the field, but it is a major problem. From our program, we keep evaluating our different lines to see the level of resistance, and as you can see on right Y bar, or the level of early blight resistance, and X bar various lines. This is just to show some of the lines. Of course, we evaluate hundreds of lines every season. So, for example, plum dandy is a hybrid, whereas NCEBR 6. 6 is one of the lines. NC1 CLBR is another breeding line. EBR2 is another line. Point here, major message of this line is that although these lines, like in addition to hybrid, these breeding lines were also released as a resistant line, the level of resistance is changing. Means they do not have same level when we evaluate multiple years, and means it is changing either because they are not they are losing their resistance, or there may be a new race. Or that means what it tells us is we should find new source of resistance. Initially, when these lines were developed, which were developed by Dr. Gardner, who retired in 2008, he used three sources basically: PI 126445, and another source was C or Campbell 1943, and 71B2. 71B2 is the main source to develop plum related lines. That is mainly that is the line that was used to develop NCEBR 5 and 6, and eventually plum dandy, which are plum, whereas the former, like C1943 and PI 126445, these are the lines they were used to develop NCEBR1 and EBR2, and eventually EBR3 and EBR4. These are large fruited breeding lines, which were eventually used to develop Mountain Supreme. And Mountain Supreme, they were the one to develop further breeding lines. We used we developed population using like NC1CLBR, which is resistant to early blight, as I showed in previous slide, which has still a very good level of resistance to early blight. Why? Because we wanted to map the QTL and also combine the QTL resistance and also Septoria leaf spot resistance, because Wisconsin 55 has reasonable level of resistance, not quite a lot. And NC1CLBR, again, same, early blight resistance, and Florida line, which is susceptible to early blight, is still it has good fruit quality, and it has some other useful traits. So we developed two independent population, and wanted to map the QTL and also combine some fruit quality traits. Using this population, we mapped the QTL. Literally, although it was like 6 QTL to me, basically, since they are close, it may be only 3 QTL. We don't know, but we identified 6 QTL, and this is already we have already reported it. Okay. And we have advanced one of the populations to later generation, like all the way up to F8 generation now, and looking at the evaluating it further. Even we evaluated until last year, and evaluating it this year now. It shows the distribution at various locations. And again, it shows the correlation between different traits in different location, different location. And now what we are doing is we genotyped the population using genotype the population using OmniSeq, which is very similar to GBS. It uses multiple restriction enzymes, and it shows on right hand side it shows the distribution of different genotype categories. This blue and green, these are homozygous, and obviously, as expected, the heterozygous number of heterozygous are very few, as you can see here, which is what we would expect. And on right-hand side, upper right-hand side, this is the density map. It shows the distribution of SNP molecular marker, and on right-hand side below, this is the distribution of SNP molecular marker on different chromosomes. Based on that, we are going to do the QTL analysis. We still have to do that. Coming to septoria leaf spot, one of my former graduate students, she started working on it. Again, septoria leaf spot is very important disease here in North Carolina and in Northeast, particular of the US, especially when there is hot and humid environment condition. And of course, the temperature is going up, and the problem is getting more severe now, and there is no any resistant line until now. So now, Jackie started working in one of the populations, and Anju is taking that project to a large extent. Why? Because Jackie and postdoc Dr. Siddiqui, Dr. Siddiqui wrapped up the project and identified some of the QTLs. We have just submitted the manuscript, and we thought that we should expand the base to identify the source of resistance. So Anju, who is the PhD student, she started evaluating more than 250 lines, including some of the wild relatives, so that we identify the source of resistance and we can utilize it to develop the resistant lines. As I mentioned before, septoria leaf spot is confusing in terms of its symptoms with early blight, but its spot size is much smaller, but number of spots are larger per unit area when we go to the field, as you can see on right hand side. Say, for example, right here. Like they have like a gray and tan color, and they are pycnidia, will can be seen in the center of the spot when we go to the field, and you can feel it when you go to the field, and based on that you can identify the disease. Right here, this is the typical symptom, and this is the pycnidia is kind of like raised when you feel it. Yeah. As I said, like, Anju is leading this project, and we have not published this data yet. And she evaluated several lines, and she's going to do GWAS. Based on that, we are expecting to identify multiple lines, and also identify the locus, conferring resistance to septoria leaf spot. Also, at the same time, postdoc Dr. Siddiqui is advancing some of the mapping population that we started developing using some other source of resistance, which is already at advanced generation now, and this shows the correlation among the multiple some of the traits. Some of the traits, as you can see here. And again, in this case also, we mapped the, we genotyped the population using OmniSeq, and on right here on the lower right, it shows the number of, type of, genotypes, and on right, upper right, it shows the density map, whereas lower left, it shows the distribution of some SNPs. It shows this slide shows the frequency distribution in one of the population. In one of these population that was evaluated in multiple location. Obviously, although it is not normal distribution in some cases, for example, this one and this slide, but depending upon the population size, it may happen. Again, this is the, so this is simply the correlation, and this is the principal component analysis. This is the heat map, again, distribution of SNP molecular marker. And based on that, QTL analysis was conducted, and we have identified QTL, as you can see here, like from chromosome number 6, 9, 12, probably, QT, probably, 6, 9, 12, and also 1, 3, 7. And this manuscript has been just submitted, and we're going, we also received the comments from the reviewer, and we're going to submit the revised version very soon. Next is late blight. Late blight is, we know that this is very important disease again. Once, the late blight, there is onset of late blight in the field, it can have, wipe out the crop within a week or so. In the field, there is very distinct differences. This is a picture from one of the organic trial. As you can see, if there is no resistance, the crop can be, infected entirely. Whereas if there is resistance, it can hold up quite well. So this slide shows the symptoms of the lead blight, how the oomycetes grows on left-hand side, whereas right-hand side, it shows the evaluation of some of the lines in the program, meaning that, we have some of the lines, they do not have, they are quite resistant, means, this is just to, so the distribution of the disease, or, in fact, these lines have different combination of like a PS2 and a PS3. And in fact, before that, I should have mentioned that like PS2 is conferred by lead blight is conferred by 3 different genes like PS1, PS2, and PS3. PS1 is not effective. PS2 and PS3, if we combine them in a homozygous condition, it can confer resistance quite well. And we have lines. They are at homozygous condition for PS2 and PS3. So for example, in this one, like it shows like the ANC25P, which has PS3. If it is at homozygous condition, it has some level of resistance. So these lines, they do not have disease or very low means. they have, combination of PS2, PS3, so on and so forth. Plum regal, which is at heterozygous condition for heterozygous for PS3, it has more disease, so on and so forth. Means if we combine PS2 and PS3, they will not have they will not have lead blight. That is the point of this slide. So we'll try to, whereas for example ANC 30P, it does not have any PS2 or PS3 which has more disease when they are evaluated at when they are evaluated at the scale of like 0 to 5. So in those PS2 and PS3 are major genes, but there are reports that there are also quantitative resistance, quantitative control, polygenes controlling conferring resistance to lead blight. So considering that, we wanted to map the QT, look at the quantitative inheritance for lead blight, and we evaluated it in multiple location, using one of the populations, and we see some we see quantitative distribution when we evaluated in multiple location, as you can see here. And in fact, like on left hand side, it shows the distribution for mapping PS2 on chromosome number 10, whereas on right hand side, this is the mapping of PS3 on chromosome number 9 when it was evaluated for like multiple years, like 7 or 8 years. It has been reported before, and there were some other minor QTL reported from other chromosome also, like your chromosome number 12 and chromosome number 12 and chromosome number 12, but its effect was very, very minor. Coming back to this slide, when we want to score this type of disease, like septoria leaf spot, bacterial spot, or early blight in the field, it may be confusing when we go to the field and start rating. So the situation is that, like in research plot, and what we want to do is we want to identify the disease accurately. That is one thing. The other thing is we want to identify the disease, precisely what disease is this, and also rate the disease accurately. So both are, very important. And because of the, confusion in the disease identification and also, rating, the current situation is that there is cost, there may be inaccurate rating, and also there is cost involved in phenotyping, because we have to phenotype hundreds of lines in the field, and also there is limited quantitative assessment. We have not been able to phenotype the data accurately. That is the situation. And also, of course, data set is not available. Considering that, the Li Rong, who is the PI in biological and agri engineering department, and her student, they are working. We are working in collaboration about like high throughput phenotyping of these disease, foliar diseases. Currently, we score disease at the scale of 0 to 5 or using like Hertzsprung-Barrett scale. In Hertzsprung-Barrett scale, like we use the scale like 1 to 12. The disease percentage is given like the, here like in this column, 0, 0 to 3, 3 to 6, so on and so forth, and it may be again, say for example, for new researcher, like if it is scale is 1, it should be, 0% infection. If it is 2, 0 to 3% in disease infection. If it is 3, 3 to 6% infection. If picture like this, what is the, level of infection, something like this. So, this is based on like level of infection and, rating, So for example, if you have any image from the field, what may be the score, that is a type of situation. So considering that, Lirong, she has this high throughput background using robotics, robot, and she started thinking about it. There may be some human error if we use, our eyes for disease rating, and we started working together, and she started, to address the issue of like time and also labor cost involved in phenotyping. If we develop some robots, that may address the issue of both, time as well as cost involved in disease phenotyping and also disease identification. By assigning the actual score. So, for example, like 1 for like no disease, 2 for this level of disease, 3 for disease, 3 for this much of disease, so on and so forth. By training the robot in actual situation, how we are doing is taking the picture by this robot. This is the robot. This is our tomato field here at Mills River. This robot takes the image from our research plot. Okay, this robot takes the image from our research plot. Obviously, they are some of those are have a disease like of bacterial spot or early blight, septoria leafy spot, and also like late blight, so on and so forth. They score different. They collect, images at different stages, and obviously they have different score, like scoring all the way from 1 to 12 or 0 to 5, whatever score we assign, it takes. And also at the same time, there is like there is there is they can I think like they can there is Unreal engine. I think they can develop the simulation, situation like this is robot on this is actual field, whereas this is simulated field and a simulated plant condition, and like simulated disease under simulated condition, and develop the image, and create the simulated data, and using that scenario assign different conditions. Say for example, they define like this, like like this is background black is black mulch, that is mulch condition. Fruit, red fruit, and a healthy plant, green, Healthy plant is green, disease is yellow. Healthy on upper panel, it is like, disease, and disease is yellow, means on lower panel, as you can see here. And what is the percentage of yellow? Based on that, like 31% leaf area, Hertzfeld-Barrett score will be about 6. Hertzfeld-Barrett around 5, 23%, 16%, 5, so on and so forth. They can assign that percentage, and by feeding that data, I mean taking images at different stages, and defining that condition, um, you know um, they can develop the model. And in fact, that model has already been developed, and it has already been published in one of the publications before. As I mentioned before, the simulation data has been developed in a specialized simulator using on sorry, this is unrealistic engine, means this is a kind of the air condition, she has better information, obviously, than me. But how, in what aspect I'm helping is using my population, breeding population, segregating population for these diseases, and also food quality and these things. And her background in robotics, we are working in collaboration, and developing models, and also like disease detection, developing disease detection algorithm, and also model for universal phenotyping system for Foliar diseases. I think this model will be very useful. Useful. So just summarizing this, overall mapping QTL, doing GWAS, and identifying the source of resistance, and identifying the genes, and developing the molecular marker. Although I did not focus in developing molecular marker, but we have already developed molecular marker for 7-8 various diseases, and we are using those molecular marker in our regular screening program, disease screening program, which which we screen in our early stage, before taking to the field. And we continue our mission to develop the molecular marker. And using this molecular marker information, and also using this high throughput phenotype molecular marker and conventional breeding, we have already developed breeding lines and varieties of different fruit types, like large fruited, plum or Roma type, and grape tomato varieties, which is here is the list of hybrids on the left hand side in the table, and breeding lines on right hand side, which have already been released. And with that, I would like to conclude my presentation. I would like to thank my granting agencies, like National Science Foundation, USDA, NCDA, and Growers Association. Dr. Gardner, who retired in 2008, and he's still working in specialty type of tomato. Reza Shekasman is a research scholar. He is working in verticillium wilt and compact type tomato. Dr. Jianbo Jangs, Irfan, and Sikandar, these are postdocs and are doing excellent job. Anju and Rajan are PhD students, and Matthew and Wayne are research technician. With that, if you have any question, I'll take it.
Thank you, very much. Thank you, Dilip. If you wanna maybe stop sharing or have several questions here, okay? This first one from Elise is talking about bacterial blight. Is QTL11 corresponding to RX 4
Yeah.
slash XV3? Is
Yes,
that
it may.
confirmed? Okay,
Yeah.
all right. So
Yes,
that's
yeah.
confirmed. All right. So the next question is from SAP, good presentation. You report some QTL with lower LODs values, 2.5, influencing particularly for septoria, leaf spot. As you
Mhm.
mentioned, the one objective of your program is to develop markers. Do you think these QTLs with lower LOD value and minor effect could be used in molecular assisted selection?
Yeah, frankly, I do not feel very comfortable with this QTL. Why? Because the source of resistance itself was not very contrasting in a sense that its resistance level was not very high.
Mhm.
So, I mean, because of that, the line that Anju is screening, we are going to develop the population, and that may be very useful from her GWAS study.
Good.
Yes, no, we may not be able to develop the useful molecular marker for marker-assisted selection from this QTL study.
Okay. Question from Pedro Belicuas in Brazil. Regarding the QTLs, do you observe any negative genetic correlation between disease resistance and other attributes like yield or fruit quality?
That is a good question. I do not have that experience, Charles.
Okay.
Uh,
Okay. It's a good question. Yes. Gustavo from Argentina is asking whether these tomato diseases that you're breeding for are endemic here in North Carolina or if you inoculate in order to do the screening.
we do both. Like
Okay.
yeah, we do both. Like septoria leafy spot and olive blight. We collect data from the greenhouse and also collect data from field condition. There
Mhm.
is very conducive condition in Waynesville. Both disease appear every year. And also for bacterial spot, we collect data from field condition where we collect data under natural condition, and also to make sure that we get uniform level of infection. Population was inoculated under field condition, but data was not collected from greenhouse condition in case of bacterial spot.
Okay.
Mhm.
Question from Lingmen Yang. Could tomato disease resistance genes, the ones you identify, be transferred to potato, like late blight?
I am not sure. That is a good question because the pathogen like Alternaria and
Mhm.
Phytophthora infestans, the pathogen is the same. I was talking with one of the pathologists, and he said that they have. Let me see what QTL. I think he said a major QTL in case of potato. Man.
Mhm.
No, I'm not sure. I'm not sure. I think PS2, PS3. I'm not sure about that. But
Mm.
the pathogen is the same.
Yes. Yeah.
Yeah, pathogen
Okay.
both. Yeah.
And
Mhm.
this is my specific question. How much work is being done with wild species as a sources of resistance? Are you doing anything in your program, or are you collaborating with people doing research?
You know, I try not to, work much, not to use much wild relatives. Why? Because it has a linkage drag,
Yes, yes.
and fruit quality is very important, and it is very difficult to minimize the improve the food quality and but in some cases we have to use wild relatives for sure. In that case we want to use only like Pimpinellifolium and Serraciforme, which are easy in crossing, and also their fruit quality is relatively better.
Okay. Another question from Gustavo. He says that in the box plot you show briefly the positive allele associated with disease always came from the resistant parent. Have you seen sometimes contribution from the susceptible parent?
Yes.
Okay.
Yeah. Susceptible parent. Yeah, in the QTL analysis. Yeah.
You have seen positive contributions from
Yes.
susceptible parents.
Yeah.
Okay. Well, I think thank you so much, Dilip, from very comprehensive presentation. Thank you for the participation of a lot of you. Sorry, I need to get here. And we look forward to having you next Thursday for the last seminar of this series by Dr. Aurelio Aguiar, who's going to talk about eucalyptus breeding in Brazil the same time at 11 a.m. So I appreciate your participation and look forward to having you with us for our last seminar of this series. Have a great weekend, and thank you so much, Dilip, again, okay?
Okay, thank you very much for the opportunity.
Okay, goodbye.
Okay, bye.